Welcome everybody to this month's roundtable. We have Dr. Kai He from Assistant Professor from the Mayo Clinic. At the end of the seminar, please unmute if you had questions, unmute yourself. I think it gives better interaction and ask your question. If you don't feel comfortable doing that, go ahead, put it in the chat and we'll ask it for you. But I encourage you to be active participants in the meeting. Dr. He, Assistant Professor from Mayo, will be telling us today about microtubule glutamylation in the primary cilia, a potential therapeutic target for AD, PKD, and ciliopathies. So Dr. He, I'm going to turn it over to you.
Okay, I'm going to share my screen now. And so do you guys can see my screen?
Go to the full presenter.
Okay.
Perfect.
Okay. So first I want to thank you for giving me this opportunity to present our study to the PKD RRC community. So I'm Kai from the Mayo Clinic PKD Center and the Department of BMB. So today I'm going to talk about the various special tubulin post-translational modification in cilia and in ciliopathy and ADPKD. So some background introduction. First is a primary cilia. So primary cilia is a hair-like organelle protruding from the cell membrane. And actually it can be formed in most type of mammalian cells. And it functions as the sensory antenna, which mediates many pivotal signaling pathway during development and tissue homeostasis. For example, the GPCR signaling pathway, HRC signaling pathway, the receptor tyrosine kinase pathway, and some other signaling pathways. Since the primary cilia has such an important signaling function and also it present in almost all the type of cells, it is not surprised that the cilia defects could lead to a wide spectrum of human diseases, which term as ciliopathy. So the ciliopathy usually manifests with multi-organ abnormalities such as the kidney, liver, and bone and fat tissues and so on. Although the cilia has such important function, unfortunately, currently there's no safe and effective drug or drug target that can be used to restore the cilia's function in ciliopathy context. So another information is about the microtubules. So you know that microtubule is a highly conserved cytoskeletal structures. It is made of alpha tubulin and beta tubulin heterodimers, which can be polymerized into protofilaments and then further form the microtubules. Although the basic structure of a microtubule is highly conserved across all the species, actually, the microtubule can adapt to a huge diversity of cellular functions. For example, the microtubule in the axon of the neurons, the marginal band in the microcryocyte, and the mitotic spindle and the mid-body during the cell cycle progression. And importantly, the axon in the primary cilia, multicilia, and flagella. So the mechanism that give rise to the functional diversity of a microtubule is the tubulin code. So actually there's two mechanisms that can generate the tubulin code. So one is the tubulin isotypes. So as you may know, both alpha tubulin and beta tubulin have many isotypes. So by the combination of different tubulin isotypes, it will give rise to the diversity of the microtubule. So another mechanism is called the tubulin post-translational modification, which is PTMs in short. Currently, there have several PTMs have been found on tubulin, including polyamination, phosphorylation, acetylation, and polyglycylation, tyrosination, and detyrosination. So we are particularly interested in this post-translational modification, which is called polyglutamination. So which means that the C-terminal tail of alpha or beta tubulin can be modified by a chain of glutamate. We are particularly interested in this modification because the tubulin glutamination in ciliated cells, it predominantly occurs in the cilia, but not in the cytoplasm, suggesting that the tubulin glutamation have a very specialized ciliary function. So this modification actually is directly modified by enzymes, including the glutaminases and the deglutaminases. So the glutaminases is responsible for formation of the glutamination chain. On some of the glutaminases, such as TTL4, 5, and 7, is responsible for the initiation step, while the others were mostly responsible for the elongation step. Well, correspondingly, there's several deglutaminases that can erase or remove the glutamination from the tail of alpha-beta tubulin. For example, this deglutamase CCP146 is responsible for removing the long-chain glutamate, while the CCP5 is the only deglutamase that can remove the branching point glutamate. But just recently, people identified a new type of deglutamation, which is TMCP1, but it's very special, have a special function. Previous zebrafish studies suggest that glutamination in cilia have very important functions. For example, in this zebrafish, when they knock down TTL6, so TTL6 is a polyglutamase in zebrafish. When they knock down TTL6 by morpholino, you can see that the glutamination in cilia is significantly reduced. And importantly, these fish will exhibit very typical ciliopathy phenotypes. For example, the body curvature and the pronephric cyst and abnormal autolysis. So another example to indicate the important function of this particular tubulin post-translation modification in cilia is the Joubert syndrome. So Joubert syndrome is one of a well-known ciliopathy, which is mainly characterized by neurobiological defects, such as the mono-tooth sign in the brain, and some other phenotypes involving the hypotonia and developmental delays, abnormal breathing patterns, and other features such as cystic kidney, polydactyly, and retinal dystrophies, and so on. Genetically, Joubert syndrome is a highly heterogeneity. Actually, the dozens of serial gene mutation will cause the Joubert syndrome. Very interestingly, actually, several of these Joubert syndrome gene mutation will cause defects in of axonal glutamination in cilia, including AR13B, CP41, KIP7, ARM-C9, and Togran. As you can see that mutation of these Joubert syndrome genes will greatly induce greater reduction of glutamination level in cilia, suggesting they have an important function. Although the glutamination defect in cilia is linked with ciliopathy, but what's the exactly function of this particular modification in cilia was still unknown. So our previous study first to want to know which kind of glutaminase or deglutamase is the major enzyme in cilia. So in short, we found that the TTL5 and TTL6 are the major glutaminase in cilia. And interestingly, this enzyme exhibit very dynamic ciliary localization pattern. Well, as you can see here, mostly the TTL5 and TTL6, this enzyme actually enriched at the cilia base without enter into the cilia. But some of the cilia, you can see the both TTL5 and 6 can be localized inside the cilia proper. And interestingly, you can even see some cilia with both patterns. And you can see both the TTL6 and the 5 can accumulate at the cilia base, but also you can see that they can be observed inside the cilia. So this pattern suggests that the trafficking of this enzyme is very dynamic and is highly regulated pathway. Importantly, when we knock down this enzyme, you can see that the glutamination in cilia was significantly reduced in terms of reduced glutamination length in the axon or cilia. We confirm that this reduction of glutamination length is not because the cilia get much shorter. You can see that the cilia itself is intact. And very surprisingly, we found that when we knock down TTL5, actually it does not quite affect the cilia formation. in terms of a cilia genesis ratio, and also does not quite affect the cilia length. And for the D-glutamase, we found that this D-glutamase CCB5 also localized in cilia. And when we overexpress this D-glutamase, it will reduce the glutamination in cilia as well, which is similar as we knock down the glutaminase. Well, when we knock down this D-glutamase, as we expected, you can see that the glutamination in cilia get much longer and stronger. And also the cilia length will become much longer as well. And also, when we knock down CCB5, it also does not affect the cilia genesis. So let's raise the question, so why glutamination is so important? So we next to examine whether the glutamination actually regulates the cilia signaling. So because in zebrafish, the TDL knockdown zebrafish has a cystic kidney phenotype. So we wonder whether it affects polycystin pathway. So as you know, the cellular localization of polycystin actually is very important for their function. As many of the PKD1 missense mutation exhibits defects in cellular localization. And also the PKD2 have a similar phenotype. So particularly in this PKD2 mutant, which is the LRM4 mutant, this PKD2 mutant still will preserve their calcium channel activity, but importantly, the PKD2 mutant failed to localize in primary cilia. And this PKD2 homozygous mutant also developed with a typical PKD phenotype. And importantly, the severity of this mutant is comparable to PKD2 knockout mice, suggesting that the serial localization of polycystin is is a major function site for the polycystin complex. So we examine the polycystin localization in cilia, particularly is the PC2. And very interesting in that when we knock down TDL5, we do observe that the glutamination become much shorter. And very interestingly, the PC2 localization in cilia also become very short. And this residual PC2 localization actually is overlapped with the residual glutamination region. And we confirm that this reduction of length PC2 is not because the cilia gets shorter. As you can see, the cilia itself cilia length itself is intact. So importantly, when we restore the glutamination in the TTO5 knockdown cell by co-depleting the D-glutamate CCB5, you can see that the glutamination is restored to normal level, and the PC2 cilia localization is also restored. So this data suggesting that polyglutamation is essential for cellular localization of polycystin 2. And we also kind of confirm that actually, because PC2 cellular localization is important for cystogenesis, so we also perform the 3D cystogenesis assay to confirm that actually the polycystin signaling was compromised when we reducing the glutamination in cilia. So here we overexpress the wild type D-glutamase CCB5. You can see that the glutamination when in these cells are very, very low. We use the mutant CCB5 overexpression as a control because this mutant CCB5 don't have those enzyme activity. So glutamination in the cell is normal. And you can see that when we overexpress the wild type CCB5, the cystogenesis is largely enhanced. And we confirm this result by knocking down the glutaminase TDO5. You can see that knocking down this glutaminase can also promote the cystogenesis in 3D cultures, suggesting that the axonymal glutamination defects promote the cystogenesis in 3D culture. Well, another example that shows that the axonyl glutamination is important for cellular signaling is the hedgehog signaling. So as you know, the hedgehog signaling is one of the most well-studied cellular signaling pathway. Actually, it's a very complex signaling pathway. It involves many signaling molecules trafficking inside and outside the cilia. So, and the GLI is one of the core component of hedgehog signaling pathway. When you activate the hedgehog signaling, the GLI2 and the GLI3 will move to the cilia tip and become active form, so and then traffic out of cilia to regulate downstream events. So from here, we can see that when we treat the cell with sodium hedgehog agonist, SAG, you can see that the glycerol will traffic to the cilia tip. And interestingly, when we knock down TTL5 to reduce contamination in cilia, you see that the glycerol cilia tip accumulation was significantly suppressed. Well, if we restore the contamination in cilia by co-depletion of CCP5, the glycerol cilia tip accumulation can also be rescued. And we also checked the hedgehog reporter gene GLI1, and you can see when we knock down TTL5, the upregulation of GLI1 will be suppressed. But if we co-depleting the CCP5 to restore the contamination in cilia, the GLI1 expression will be partially rescued. Or another example to kind of highlight the contamination is important for cilia signaling is the BMP pathway. So BMP pathway is a very conserved signaling pathway, which is important for kidney development and bone development. Well, this pathway it is initiated by the ligand binding to the receptors, which will activate its downstream effector SMAD protein by phosphorylation. So when this SMAD is phosphorylated, it will further be translocated to nuclear to activate downstream gene active transcription. So one of our recent science transformation medicine study actually discovered that primal cilia is very important for BMP signaling transduction. Specifically, we found that the receptor of BMP pathway and other key component of BMP signaling pathway localized on the primal cilia. And this cilia-mediated BMP pathway is essential for the chondrogenic differentiation and osteogenic differentiation of mesenchymal stem cell during the muscle injury and repairment. Very interestingly, when we decrease the contamination in cilia by knocking down TTL6 or overexpression of CCP5, the BMP signaling pathway also suppressed significantly. Actually, our recent data suggesting that contamination in cilia is kind of a general regulator of signaling pathway, as it also regulates other signalings, including the receptor tyrosine kinase signaling, TGL beta signaling pathways, and other signaling pathways that it can be mediated by cilia. So this evidence kind of give us a very strong conception that actually the axonal contamination is a key regulator of a ciliary signaling. So since the contamination is important for cilia signaling, and another factor is that contamination is indispensable for cilia formation. So it will suggest that the contamination in cilia actually a very good target for manipulating cilia signaling. Well, as a drug target, one of the key questions is that whether this drug target is targeting this pathway is safe or not. Actually, the study from the CCP5 knockout mice suggesting that if we increase contamination in cilia, actually it's very safe because CCP5 is a deglutamase. When you knock out this enzyme in mice, the contamination in cilia will be strongly increased. But the CCP5 knockout mice actually do not exhibit overall abnormality. So this evidence suggests that increasing the axonal contamination could be a safe strategy for enhancing the ciliary signaling and functions. So this data kind of strongly encourage us to kind of identify some small molecule or drugs that can promote the contamination in cilia. So with this idea, we perform the kinase inhibitor screening to aiming at to find some drugs that promote contamination. So here we use the RPE cell as a model because this cell have very consistent cilia in length. So we first use cilia length as a readout because if you increase contamination, the cilia will become much longer. And then we further want to exclude them contamination independent effect of these drugs using the hypoglutaminated cell models. So particularly, we use the TTL file knockout cells and the CGP file overexpress cells, and see if those drugs can restore the glutamation in these cells without affecting the cell length. So in short, we identify several positive hit compounds, and this drug called abemaciclib is one of the top hits. So abemaciclib is the CDK4 and 6 inhibitor. As you can see that the abema can significantly increase the length of a glutaminated axonine. Also, the CR become much longer. And we also validate that the drug promote CR length is because it specifically promote glutamination CR. So we use the TTLL file knockdown cells. So you can see that in the TTL file knockdown cells, the glutamination will become much shorter. But if you treat the cell with abema, the glutamination in the cells is completely restored. And the CR length is without affecting the CR length. So because this drug can inhibit both CDK4 and 6, so we wondered actually which of those target of this drug is responsible for regulating the glutamination. So we knock down the CDK4 and the CDK6 individually by specific siRNAs. So, and interestingly, we found that only knock down CDK6, but not knock down CDK4 can promote glutamination in cells, suggesting that it's the CDK6, but not the CDK4 suppress the axonal glutamination. Well, we also figured out what's the mechanism of CDK6 in inhibiting the glutamination. So as I mentioned before, the TTL file and TTL6 mostly are enriched at cilia base without enter into the cilia. But when we knock down CDK6, you can see that the majority of cilia can be observed with TTL file and TTL6. So suggesting that the CDK6 can suppress the cilia import of this glutaminases. But since abemaciclib as a drug can promote glutamination, so we wonder whether this drug can be used to enhance cilia signaling in the ciliopathy cells. So here we use the Joubert syndrome as a ciliopathy model because in Joubert syndrome, the glutamination in cilia is compromised or significantly reduced. Specifically, we choose these two Joubert syndrome genes. One is the RMC9, another is a CP41. As you can see that when we knock down RMC9 or CP41, the glutamination in cilia is significantly reduced. And also the Hedgehog signaling also significantly reduced as indicated by the impaired cilia tip accumulation of GLI3. Well, importantly, when we treat the cells with abemaciclib, you can see the glutamination can be restored, and the cilia tip accumulation of GLI3 can be also rescued, suggesting that the abemaciclib can rescue the Hedgehog signaling pathway. And also the abemaciclib can rescue the polycystin-2 localization in Joubert syndrome cells. Well, you can see that when we knock down RMC9 or CP41, the PC2 cell localization was significantly compromised. But you treat the cell with abemaciclib, you can see the polycystin-2 cellular localization can be restored. Well, in summary, our study demonstrated that glutamination in cilia is important for cilia signaling. Particularly, it is very essential for polycystin cellular localization, and also very essential for GLI2 and GLI3 cilia tip trafficking, and also also important for other cilia signaling molecules. And also we found that at the cilia base, the CDK6 can inhibit the glutamination by inhibit the cilia trafficking of the glutaminase get into the cilia. Since the CDK6 can regulate the cilia glutamination and thereby regulate cilia signaling, including polycystin signaling, so we was wondering whether actually CDK6 is involving in ADPKD. So we first examined the expression level of CDK6 in ADPKD tissues in the kidney tissue. As you can see that actually the CDK6 is highly upregulated in PKD tissues, kidney tissues, especially in the cistern lining cell in human kidney tissue or the mouse kidney tissues. So because the CDK6 is an inhibitory regulator of glutamination in cilia, so we further wondering whether actually the glutamination in the PKD cell also was affected. So we use a PKD cell model. Specifically, we use the PKD1 RC plus cells and the PKD RC minus cell. So this is an immortalized collecting duct cell that isolated from the PKD RC plus kidneys. And this cells is established by Vishal Patel's lab. And the PKD RC minus cell actually is derived from the PKD RC plus cells. We use this cell because these cell types kind of faithfully recapitulate the major PKD phenotypes in vivo, including increased cystogenesis and other molecular alterations that correlated with PKD, such as mitochondrial abnormalities and proliferation pathway activations. So in short, we observe that compared to the PKD1 RC plus cells, the PC2 localization in PKD1 RC minus cells was significantly reduced. And very interestingly, we found the glutamination in cilia also reduced in the PKD RC minus cells. And importantly, when we treat the PKD1 RC minus cell with abemaciclib, you can see the glutamination level was largely restored. And also the PC2 cilia localization can be partially rescued. So this means that the abemaciclib can be used for PKD treatment. So actually the abemaciclib is a FDA approved drug for breast cancer treatment. It's a new generation of a breast cancer drug, which have many advantages. For example, it is orally available and is applicable for long-term treatment. And importantly, it only have very subtle side effect. Recent clinical studies suggest that the abemaciclib is very safe for renal function. As you can see that even use high dosage of abemaciclib, the eGFR level in the patient is still unaffected, suggesting the drug is very safe for renal function. So this data suggests that the abemaciclib is a good candidate for drug repurposing. So we evaluate the therapeutic effect of abemaciclib in the PKD RC mice model. Specifically, we use the F1 PKD1 RC from the F1 generation of PKD RC mice with the background of BL6 and 129 background. We use this particular mouse model because previously Peter Harris and Dr. Katharina's lab demonstrate this F1 generation PKD RC mice develop PKD very rapidly, but they only have very small variation in kidney cyst, so making it a very good model for preclinical trial. So we also we first conduct the early intervention study by treating the mice at 4 weeks old. And we will randomize these mice by total kidney volume to make the different group well matched in terms of disease severity. And the data suggests that the drug shows very good therapeutic effects. And you can see from our MRI image data, the 10 weeks treatment of abemaciclib is already show very good therapeutic effect in terms of TKV reduction in a dosage-dependent manner. And because the PKD patient usually need to use the drug for very long term, so we kind of further to evaluate the long-term effect of the drug treatment to extending the drug treatment to 20 weeks. And you can see from our data, this abemaciclib also have a very good therapeutic effect for 20 weeks treatment. And very importantly, this drug does not quite affect the body weight of the mice, suggesting the side effect of the drug is very low. Here is a more kind of detailed data to indicate the therapeutic effect of this drug in terms of kidney weight to body weight ratio, CC index, and very surprisingly, this drug can significantly suppress the fibrosis of the kidney. And also it can reduce the BUN level, suggesting that the drug can preserve the kidney function. And also because the PKD patient usually diagnosis with the disease when they are already entered to the very advanced disease stage. So to make our study more clinically relevant, we also conduct a late intervention study. So we treat the mice at 14 weeks old. Because this at this stage, the mice have very severe advanced PKD. And we also kind of randomized these three mice in 3 different groups by total kidney volume to make sure the disease severity in this treatment group are the same. And we divided the mice in 3 groups. One is 14 weeks. Another group said let the mice without drug treatment for other 10 weeks, or treat the mice with drug for 10 weeks. Here we use a little bit higher dosage of the drug. And the data shows very promising. You can see with the drug treatment, the cystic phenotype is kind of comparable to the 14 weeks old mice. So, and in terms of cyst index, and very importantly, when you look at the kidney weight to body weight ratio, actually the mice with drug treatment is even lower than 14 weeks old, suggesting that it may show some disease reversal effect, especially in the male mice. And it also, you can see that the BUN level is comparable as 14 weeks old. But for the fibrosis, it seems that this drug does not quite reverse or reduce the fibrosis process, but it preserves the kidney function very well. So all this preclinical trial data are very encouraging us to find some additional drug that can promote contamination, and supposedly it can be used for PKD treatment. So actually, by our drug screening, we also identify some other kinase inhibitors that can promote contamination, and other drugs can even reduce the cell length. We are quite interested with this particular drug because this drug can specifically promote the contamination in cilia without affecting the cytoplasmic contamination. And also, this drug has shown very low cytotoxicity, so making it a very good candidate for preclinical trial of drug repurposing. And we confirm that this drug can suppress the cytogenesis in 3D cystogenesis cultures, so suggesting that drug can be used for potentially can be used for PKD treatment. Besides some drug that promoting the contamination, we also found some drug that can reduce contamination in cilia. For example, this particular one, DOK inhibitor. You can see that this drug treatment can reduce contamination in cilia, but does not affect the cell length. And when we look at this drug's target protein, which is DOK1, we identified that actually this protein can localize in cilia, suggesting that it is a novel ciliary protein which can regulate the contamination in cilia. And besides the drug screening approach, we also use a proteomic strategy to aiming to find some new regulator of contamination. Specifically, we use the bioAD assay. So BOR-A is a biotin ligase, which can biotinate the protein that nearby them, that within the 10 nanometers. So when you fuse BOR-A with a protein of interest, you can identify the potential interactors of these proteins nearby. So we use CP41 as a bait because CP41 is specifically localized in prime cilia. And then we think that if we fuse CP41 with BOA, it's very likely we can identify some regulator of tubulin contamination. And another reason that we choose CP41 is that CP41 is specifically regulate contamination in cilia, but not other tubulin post-transit modification. As you can see that when we knock down CP41, it will reduce contamination in cilia, but not affect the acetylation. So we kind of established the CP41 bio-ID cells and using the MIC-BOA as or expression cell as a control. You can see that the MIC-BOA CP41 can localize in cilia and also can biotinylate in the protein inside the cilia. And although there are some kind of leakage at the cytoplasm, but it still be able to localize in the cilia and label the cilia protein. So we kind of confirm that this cells works pretty good in terms of biotinylation proteins. And importantly, overexpression of CP41 does not affect the cilia genesis or the cilia length. So this gives us a good cell model to kind of avoid any artificial effects because of the cilia get much longer or cilia become more. And the proteomic data turns out very successful because we already be able to kind of identify many serial proteins in the proteomics. And importantly, on the top of the candidates, we already identify some known serially glutamation regulators, including the D-glutaminase and CSSAP1, which has been known as a regulator of TTL, and KIP7, which is a Joubert syndrome protein that regulates glutamation. So this kind of gives us very confident that this proteomics is able to identify new regulators of glutamation. So here is some example that the CP41 interactor protein is this protein called C55. Actually, we found this protein also localized in primary cilia. And very importantly, when we look at the subcellular localization of C55, you can see that actually the C55, which is in red color, actually directly interact with the TTL5, which is the enzyme. And importantly, you can see that the C55 is proximate to the glutaminated tubulin loci. So based on this localization pattern, we kind of propose this model. So C55 actually can interact with microtubule, and C45 then can recruit the enzyme to the microtubule, then mediate the glutamation on cilia axoneme. So now we are in the step of a functional validation of the identified new regulator in using zebrafish. We use zebrafish because the zebrafish can faithfully recapitulate the ciliopathy phenotypes in human, such as hydrocephalus, heterotaxy, sclerosis means body curvature, and of course the cystic kidney phenotypes, such as resemble as the PKD. Another reason we choose zebrafish because the zebrafish model is a very quick model. So using the CRISPR-Cas9 method or technique, we can observe the phenotype within 2 days or 3 days after the post-fertilization. So it's a very good model for like a big screening purpose for functional validation. So summary, our study suggests that the axonal glutamation is very important for cilia signaling. And if we increase the glutamation in cilia, can restore the cilia signaling function in ciliopathy models, especially in the Joubert syndrome model and ADPKD models. And we also found a new pathway regulating the glutamation, which is mediated by CDK6. Specifically, the CDK6 can suppress glutamation. And we also found that the CDK6/4 inhibitor, abemaciclib, it's very likely can be repurposed for ADPKD treatment. And we also identified several new regulators of axonal glutamation, including DLK1 and the C55. So we still have many remaining questions need to be addressed. First is that we still don't know how the X-animal glutamation regulates the cilia signaling. And secondly is that whether the glutamation actually is reduced in ADPKD in vivo. Although we see this phenotype in cell models, but we still don't know whether it actually happened in real in vivo situation. And the follow-up this question is that whether what's the rule of X-animal glutamation defects in ADPKD pathology? And lastly is that what's the mechanism of abemaciclib in suppressing ADPKD? Because abemaciclib can inhibit both CDK4 and CDK6. So those kinases actually have multiple substrate. So to what extent this drug suppressing ADPKD is from the ciliary aspect will still need to be investigated. So lastly is I want to thank you all the lab members in our lab and our collaborators in Mayo Clinic and other collaborators for sharing the important reagent. And also I want to thank them our funding support, especially the PKD Foundation and the NIDDK's funding support. And thank you. I'm happy to take any questions.
Dr. He, thank you very much. This is now open for discussion. So please raise your hand, unmute yourself when you're called on. I'm going to start off with a quick question. You show that changes in glutamylation alter the distribution of proteins within the cilia. Do you have any idea the mechanism of how that occurs? I mean, is it, there's the inversing compartment and so forth. Does it change a structural unit in cilia?
That's a very good question. Actually, we still don't know the mechanism, how it regulates the distribution of signal molecules. We hypothesize that it may regulate some adapter protein between the axon and the cell membrane. And one of the potential mechanisms that because the glutamine is negative charged, so if you have polyglutamation on the cilia axon, actually create the surface of highly negative charged surface. It may affect many protein binding. So, and you mentioned that the inversing compartment, actually, we're also very interested with that part because this residual signaling glutamation is kind of overlap the inversing compartment. And very interesting, no matter how we kind of knock down those enzymes, for example, when we co-deplete the TDL5 and 6, this residual signaling is still preserved there, suggesting it may have some other inversing correlated function. But structurally, I don't think it will affect the integrity of axonal or cilia structure, but particularly regulate cilia signaling.
So just to be clear, when you change the level of glutamination length, the inversing compartment changes with that? So inversin
no, we don't know whether the inversing compartment is effective or not. But I just want to emphasize that this region, glutamination region, even when we deplete the glutaminase, this region still have very strong glutamination, but the distal part will disappear. Well, we still don't, I still don't know why this happened, but it is very common in other cell models.
Okay. Questions from the group? I'm going to continue to ask questions then.
Okay.
So for the CDK6, do you think that a glutaminase or D-glutaminase, I guess might be the case, is a direct target for the kinase activity, or is it an intermediate that goes on?
Actually, we think it's kind of indirect activation. So here's a model to kind of explain what happened. So this enzyme need to traffic into the cilia. So CDK6 actually inhibit the trafficking of this enzyme by phosphorylating this particular molecule called Phe. So Phe is very important for mediating the trafficking of this enzyme get into the cilia. So if this protein is phosphorylated, the trafficking pathway will be blocked.
Okay. So that'd be interesting to combine with your BRA studies to look at the difference in the proteome under those two conditions. But
yeah, exactly.
Questions?
Can I have a question? I don't know how to raise my hand.
Hi, Dr. Chen.
After 10 years of this, you still don't know how to do it on Zoom.
After AI, no longer. All right. So Kai, thank you so much for your great talk.
Yeah.
So I just have maybe a technical question. When you actually assess the length of the cilium, you use R13B. But you say the glutamate, how do you call it?
Glutamation, yeah.
Glutamation of the cilium is shortened. Right, but how do you know that the what you call microtubule is actually formed intact?
Because yeah, very good question. Because we use other marker like acetylation.
Oh, you did that. Oh.
Yeah, the acetylation is not affected. So
okay. Oh, okay.
So the axon itself is not become shorter for sure. Right. Yeah.
Yeah. Then the second following up question is: have you actually found any membrane protein, the cilium, which is still present in the distal region in your knockout cell?
Yes, exactly. Many other, I think maybe like smoothen is one of the example. So smoothen still preserve their cell localization on the whole cell membrane, but particularly the PC2 is kind of show this truncated phenotype as it show here. Yeah. So it's very specific for several types of membrane protein and also some soluble protein as well.
Okay.
Yeah.
So this is a selectivity. Some proteins affect.
Yes, some kind of a selectivity, I would say.
That's interesting. Thanks.
Yep.
Other questions?
I have a quick question.
Ma'am, yes.
Yeah, really nice talk, Kai.
Thank you.
So you show that glutamylation is required for PC2 localization. What about PC1?
Yeah, well, good question. Actually, one of the big hurdle for PC1 cell localization is we don't have any good antibody for that. But we believe that PC1 is also a compromise based on our preliminary data, and also because PC1 localization is dependent on PC2 localization. For PC1 localization is dependent on PC2, right? So if PC2 cannot get into the cilia, it's very highly likely that PC1 cannot get into the cilia as well.
I think you need to talk to Fung about an antibody. He has some wonderful antibody resources that are working.
Okay, that's great.
Particularly in human.
Oh, okay.
The RPE cells would be a wonderful model for testing that.
I agree.
We also have some like PC1 flag tag, the flipping cells, and I think it also works pretty good. But yeah, we're using that cell now.
But Fung has characterized some wonderful antibodies. So
good. That sounds good.
Dr. Gemino.
Yes, here we go. Thanks. Quick, Kai, very nice talk. Quick
Thank
question.
You.
So, you know, the effects that you've shown with PC1 and the RC mouse, you know, this kinase inhibitor could have a lot of different effects. It might be through glutamylation. It might be doing other things, as you know. And the RC, or you know, the RC protein, you know, has got a folding problem. So, how do you think about this factor of enhancing glutamylation and therefore enhancing ciliary signaling in the context of the cilia-dependent activation factor that Samlo describes? I would have predicted that enhanced glutamylation that enhances ciliary signaling would have promoted cis growth.
Well, actually, I think maybe it's a two way to explain that. One is that kind of enhancing some signaling, cilia signaling, such as polycystin dosage. And another way, maybe it can suppress CDCA. We don't know, because glutamylation increase maybe suppress some, also have some suppressive effect, such as CDCA effect. We still unclear what's the mechanism of this drug. But from our like a 3D culture assay, we think that as shown here, if we using the genetic way to specifically reduce glutamylation, is indeed promote cystogenesis in 3D culture. But yeah, you are right. We still don't know what the mechanism.
Yeah, thanks.
I can have another question. Just back to the baseline here. When you look at the length of the glutamylation signal or the post-translational modifications under a normal condition, is the size kind of set? So it's say 2 microns, no matter how long the cilia are, or is it proportional to the length of the cilia? How's,
Yeah, the proportion is also changed. As you show, yeah, I have data right here. So you can see that when we knock down the glutaminase TDFL, actually the ratio, the glutamated length to the cilia length ratio is reduced.
reduced? Okay,
Yeah. So if you treat it with the drug, actually the ratio can be restored.
that's cool.
Uh,
So somehow it's measuring that ratio.
yeah, the ratio is more important.
Cool. We got a few minutes yet if there's any more questions. Not seeing any. Dr. He, I want to thank you for a wonderful talk. I encourage you to reach out to us if we can help in any way with your studies. For example,
Okay.
PKD1 antibodies,
Okay, sounds great.
cell lines, or whatever you want. Please.
I'm happy to.
Yeah, please interact with us, and we're here to try to help push your research forward.
Sounds
So
good.
thank you again for a wonderful talk. Terry, I don't know if Terry is on. I think this is the last one for the summer. Is that correct, Terry?
Correct. This is the last roundtable for the summer, and we'll be
Okay.
reconvening after that
All right.
in September.
Encourage everyone to register for the PKD FASEB conference. I think that's still open, Terry, maybe?
I think it's still open.
Okay.
Okay.
So if you have a chance, please do that.
Okay.
And thank you again. And it's great seeing everybody. Have a great summer.
You too. Have a good summer. Bye.